Hello, and a very warm welcome to today's Cell and Gene Therapy Insights panel discussion, titled Mastering the Journey from Initial Method Transfer to Successful Lot Release for Cell and Gene Therapies. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today are a panel of experts who, among other things, will discuss how to avoid common challenges in the journey from method transfer to lot release. As always, do feel free to pose your questions to our panel using the Ask a Question box at the bottom of your screen, and we'll try to get to these during the session today. If you would like to direct a question to a specific panelist, please try to indicate that in your message. And so now I'd like to briefly introduce our moderator for today. Dr. Audrey Chang is the Executive Director, CMC Scientific Advisor at WuXi Advanced Therapies. Dr. Chang has over 28 years of government and industry experience in conducting biological product testing and in managing laboratories. So without any further ado, I'll hand it over to Audrey to introduce the rest of our panel for today.
Thank you, Abby. Can you hear me all right?
Yep, loud and clear, Audrey.
Hello, everyone. Welcome to our panel today. As Abby said, I am Audrey, and I'm here with WuXi Advanced Therapy, here located in Philadelphia, Pennsylvania. And with me today are my three esteemed colleagues, Melissa McDonald, our testing product director, who heads up our project management and client services department. Next, we have our chief quality officer, Jennifer Chung, who's been with the company for 4 years. And rounding out the panel is Dr. Heather Malicki, the executive director for analytical development and quality control. And she sports a 17-year tenure with the company. So as Abby indicated, today's topic is mastering method transfer for a successful cell and gene therapy release program. So let me start with you, Heather. In your years of experience, what are some of the best ways to approach a successful method transfer? And perhaps you can share some of the lessons learned.
Sure. Thanks, Audrey. So definitely there are many steps to ensure that you have a successful transfer, whether the transfer is within two companies or even from site to site inside the same company. Several different things will apply, but really when I think about it I focus in on what really ensures the success, I would say it boils down to a very well-written method. So the SOP really needs to be very clear. Sometimes an expert will write it and not include all of the details that maybe a more junior scientist may find helpful. So having that well-written assay procedure is first, and then that creates the foundation of what I would say would be an important next step, which would be a gap assessment. So really for those two laboratories to think about the differences, whether there's different equipment, different reagents, different processes or SOPs that we need to think about. So an example of that could cause some challenges could be cell counting. So for instance, maybe one laboratory is using an automated cell counter, but the other laboratory is using a hemocytometer. So things like that, little details could be really important. So when the SMEs at each site or subject matter experts sit down, they should go through the method and think about those differences, and then also document those in that gap assessment. So it should be a documented approach as well as an agreed upon approach before you even start any wet laboratory work. So based on my experience here, I've seen, challenges over the years of things like I mentioned, differences in cell counting. Maybe differences in pipetting. Someone's reverse pipetting, someone else is not. Differences in cell culture plates, something that you might not think, would cause a big difference, but it really can, as well as dilution plates, interpretations of the procedures and the way the method is written. So all of those things really are important. So the final thing I'll leave you with that I think is also important to consider is, if possible, and it might not be possible, but if it is, if the new lab can actually go into the transferring lab and have the subject matter experts perform the method and have hands-on training with the new lab. I think that's really important because sometimes when you're going through the details of that procedure, they may miss certain things, and it'll give the opportunity to the new lab to kind of translate what they're seeing, as well as that the way the written procedure is. So I think that hands-on approach is also helpful when it's possible. So that's it for me. So I'll turn it back to you, Audrey. Thank you.
That's great. Thank you, Heather. I mean, one can really see that the devil is in the details and that
Yes.
the importance of paying attention even to the smallest one is necessary to ensure success. Thank you. But equally important is that regulatory agencies worldwide expect that this method transfer and subsequent validations are executed in a controlled manner and turning it over to Jennifer now as a leader responsible for quality at WuXi Advanced Therapy, what do you look for in a method transfer package, again, from your quality point of view?
Thank you, Audrey. Thank you for the question. So my recommendation is always to start with well-recognized guidance document, like ICH-Q2, Validation of Analytical Procedures, and USP 1224, Analytical Method Transfer. As method transfer is a very critical part of analytical method transfer lifecycle management, and the appropriate strategy to utilize, whether it is a partial, full, or co-validation, would depend on the complexity of the method and the competency of the receiving laboratories. The key point to pay attention, even before authoring the transfer protocol, is to determine what strategy to employ, what parameters to challenge, the number of runs that needs to be performed to gather data for statistical evaluation, how to determine acceptance criteria, and what constitute comparable results between sending and receiving laboratories. Because we are trying to prove comparable performance, I cannot emphasize enough the importance of sample selection. Together with standardizing those factors we have control over, like equipment, consumables, and reagents, as Heather already mentioned. What we want the transfer to reflect is the potential difference between laboratories, to determine if the difference observed is within the assay variability that has been experienced from the sending laboratories. A robust method transfer package from a quality and regulatory standpoint should include the gap analysis, detailed test method, as well as the associated procedures, the transfer protocol and report, the training, as well as any deviation that was encountered during the execution of the transfer, and putting all this together in a comprehensive package that are ready to support submission and post-approval changes. Thank you. Back to Audrey.
Thank you, Jennifer. It is clear that quality needs to be built in comprehensively, including all of the ancillary supporting units in addition to the method. And after all, we need to be able to fully rely on the data that is generated to make these critical decisions for these life-changing therapies. Okay, great. So now we've gotten a clear picture of what is involved from both the science and the quality perspective for a successful method handover. But I think at the end of the day, we all know that this is a people-driven process. So now I turn to Melissa. Melissa, it goes without saying that this is not a one-size-fits-all approach, and that effective communication between both vested parties is key. Can you share with us some of your project management best practices that you've seen deployed? And again, what are some common pitfalls that we all want to avoid?
Sure, great question. So there are a couple communication tools that we really use at WuXi and find to be best practices and help the team avoid pitfalls along the way. Starting off with having an organized kickoff meeting where we review the project scope, we align on the preliminary timeline, that's the plan we'll follow. We confirm the ways we'll work together, who's on the team, when they'll meet, things like that. At WuXi, we assign a specific project manager to method transfers on any custom scope. So the client has one point of contact. This person hosts the kickoff meeting and the routine connect meetings. They track the progress to the timeline. They connect the client as needed with any member of WuXi to help them move the project forward. When we're running custom projects, we are receiving results and learning along the way sometimes. So the plan sometimes changes, and we need to be ready for that. So spending that time up front for that alignment between the client side and WuXi side, it really saves the project team time during execution when the timelines are more critical and the team, is busier. Publishing a communication plan as one of the key tools that identifies the key point of contacts, the subject matter experts, escalation contacts, the right people to make key decisions is really critical at the project kickoff. Because then when time-sensitive decisions need to be made during the project execution, the team can grab the communication plan, know exactly who to go to, and the key decision makers can be involved, and then the team can quickly adjust the plan if needed and move right back into execution.
Thank you, Melissa. It's very clear. I mean, those of us that have been in this business for a long time, it's very clear that in planning for success, you have to anticipate the unexpected and have a clear plan on how to keep moving forward. Okay. Final question that I want to address from me before we turn it over to our audience that I want to address to all three of you. So say that I'm a customer and I have developed an R&D product-specific method. I want to hand it over to Advanced Therapies for transfer as well as GMP lot release testing. So what advice can you give me from the very start to set the stage for a successful method transfer? And so let's reverse the order this time. Let's start with Melissa, and then we'll go to Jennifer, and then we'll have Heather close. So Melissa?
Sure. So planning actually starts even before the project kickoff, where the WuXi business development team is gathering the client scope, the critical end dates and deliverables, the material requirements are shared, and the client provides us the information when that material will be available to start the study. Including these planning details in the quote really allows that smooth transition into the project kickoff meeting, where the project manager from WuXi can present a timeline that already includes these details. So you start off with more of a confirmed plan. Ensuring we're aware of the client critical end date and deliverables is the ultimate key input here. This helps us evaluate the timeline throughout the project. If there are any changes to the plan along the way, WuXi's project manager can give the client and present options that are most aligned with what the client needs, and we can collaboratively confirm a path forward really quickly.
Fantastic.
Great.
Jennifer, yeah.
No, thank you. So my turn. So I'm trying to keep it at very high level. So even though in the GMP world, we always say, when it's not written down, it does not happen. But in my own experience with a number of years dealing with method transfer, I always noticed that, the method are written with the many assumptions, right? Especially assumptions that whoever execute that method know exactly what needs to be done. So a lot of tribal knowledge is not included. So, as a advice is to the receiving lab, ask as many questions as you need to, because that is important to ensure that you fully understand how the method should be executed. And also very important aspect for any transfer is information sharing, including what challenges that the sending lab has been seeing with this method, as well as sharing the most common root cause failure for any assay, either validity failure or assay achieving inconclusive results. So with the mindset of transparency and openness to discuss information and sharing between sending and receiving laboratory is a critical aspect for a successful method transfer. Now I'm going to turn it to Heather. Maybe she can talk a bit more about the technical aspect.
Sure. Thank you, Jennifer. So I certainly want to build on what Jennifer had indicated. It's definitely important to go through the method in detail and ask a lot of questions, and all of the information sharing is obviously extremely important. So I think the other part is if data can be shared. That gives the opportunity to the SMEs to go through the data in detail, but also thinking about the analytical roadmap and thinking about not only the success of the initial transfer from lab to lab, but also understanding how is the method going to be used long term? Is it going to be used as a characterization assay? Is it going to be used for in-process samples, stability, release, and laying out that plan from the beginning? So don't forget about that part, even though obviously you want to get through the initial comparability between the labs. But I think having that detailed roadmap is also going to ensure the success long term of your transfer. So I'll turn it back to Audrey. Thank you.
Love this. Love all this. So now we come to the Q&A, and I really encourage, we actually designed this so that we had enough time for people to submit questions. And I really encourage you to submit questions. You've got the top-notch experts here, all in one place. You've got the science, you've got the quality, you've got the project manager. So please go ahead and ask your questions through the little box. I'm sure you know how to do that. And also, for questions that we don't get to, we will answer your questions. So please don't be shy. Go ahead and ask your questions. So looking at this one, I have one for Melissa. Melissa, it's a question said, we get this a lot. If we're transferring an assay to you, what is the typical timeline?
Yes, excellent question. So each timeline is actually custom considering the scope, the client's critical end date, when the material will be available to start the study. At WuXi, we really have to be flexible to support the specific needs of each client. So we work together with the client to pressure test that timeline and scope to execute efficiently, deliver critical results with high quality. If anyone on the line is interested in the timeline for their potential study, we can quickly connect you with the WuXi Business Development team who will issue a quote, and it'll include that estimated timeline.
Super.
Thank you for the question.
Here's one that I love. Probably a combination of Heather, well, all three of you, but let's start with Heather. Should the QC team be involved in early assay development, or should they only be involved with the more mature assays that are almost ready for transfer to QC? I love this question. I'll start with Heather.
That is a good question. I like that too. We've done it both ways in the past, but I think where we at WuXi have found it to be more successful is to start with transferring the assay into the analytical development space. The analytical, even if, to be honest, even if it's a validated, robust method, the analytical development scientists are trained to really try to drill down into that method and kind of vet out the method. Also here at WuXi, we tend to write most of the protocols and transfer, if new assays that we're developing and designing. So we write a lot of methods. So typically working with the analytical development scientists here, the method will come in, the SMEs will discuss, questions will go back and forth, and then we typically will run the assay several times in the analytical development space. All of our instruments are actually GMP, so it's very similar to the QC environment, but we run the assays, see what kind of troubleshooting, what problems, what extra questions that we have that arise through the process of kind of vetting out that method and testing it. Then the data is typically shared with the transferring lab, and it gives us an opportunity to say these are problem spots, or these are problems or areas that we really need to dig into and maybe redefine or change the wording a little bit. So once the method really kind of passes the pressure test, I like that word, I'll use that too, kind of pressure test the assay, that's when we typically we'll write it all, we'll put it in an SOP or a test method format, we'll have it ready, and then that's when we involve our QC scientists. So then depending on the needs and how difficult or different the assay is from what we already run, we may do classroom training first and then hands-on training. And so our AD and our QC scientists will typically work hand in hand in the QC laboratories and ensure that the training, is very clear, and the QC staff have opportunities to run the method, and then we'll go into whatever the next step is for the project, whether it's a qualification, maybe it's a late phase validation. And then you still have the support of your AD staff. So we found that that is a successful approach, and that's typically how we do the transfers here at WuXi.
Yeah, I agree with that. I mean, I think what you're saying is that it's important for both parties to have a scientific understanding. It's just not just a handoff of a method that is a cookbook, but actually having the scientists on both sides have a strong understanding.
Yeah, thank you.
Yeah. And just to add on to what Heather had just said, right, that the benefit of having AD to actually evaluate the methods is actually also building up the knowledge management piece, right, which is very important in ICH Q10, right? Very important aspect that as a receiving that we need to have the capability down the road, right, to be able to troubleshoot, or even looking at any possibility of continuous process improvement. So having the AD scientists understanding, knowing how to run the method, and designing a method that is fit for QC purpose, and then move into QC, I think is a key for success. Thank you.
And then I would even expand that maybe to Melissa. I mean, how are your project managers, making sure they have an understanding, at least a working understanding of the science behind the method?
Yeah, absolutely. I think I covered it a little bit earlier, but that kickoff meeting, we love to see the overview, right? To really share, encourage the client to bring their presentation. Everyone is really passionate about the impact to patient. That's really fun for all of us, including project management, to get motivated, ask the questions, look at a diagram of what's going to be transferred, and also start to write down our questions or where we see as project managers, we can connect the right people, right? Where there might be parts of the project that there's some discussion that wasn't fully resolved during the kickoff meeting. We make a note of that. We say, who are those two SMEs? I know that I'm going to be having an ad hoc meeting, planning an ad hoc meeting for those two later, right? That's really the part where we get the team motivation in the kickoff, and also do some behind the scenes project managing work to be ready for what's going to come later.
Okay, keep the questions coming. They're good. Here's one that we can have a 2-hour webinar on itself. But it's a good question. How should we approach potency assay transfer? Dr. Maliki, this is yours.
I figured you would call on me for this one. Yes, like you said, definitely a 2-hour webinar for potency assays. Potency assays, I think that the inherent challenge is when you say potency assays, it could be anything from a biological assay, it could be a cell-based assay, it could end up having various endpoints. I've seen anything from HPLC, Western blots, ELISAs, all different types of assays. So that being said, potency is kind of a broad terminology. But what I would say for a successful transfer is really having, again, and not to go into it again, but a detailed procedure. But anytime you have a cell-based assay and various endpoints, whether it's ELISA or a flow-based assay or anything like that, patience, you need to have patience. You also really need to think about the cells. So for instance, are you going to have two different cell banks and try to show equivalency, or are you going to make one large cell bank, which I would recommend, make one large cell bank, try to split that between the two laboratories, try to standardize as much as possible anytime you have a cell-based potency assay, because they are very variable, they're very challenging. So any place that you can minimize the variability or the variations, that is definitely going to be the best. I know it might be difficult to have enough material, but that's where I would start. And then also thinking about the controls. Do you have a representative control? If it's a viral vector-based potency, having a representative lot. If you're early on in development, it might be a PD batch. But as you you go through the clinic, I would highly recommend replacing that to ensure that whatever your reference is similar to what you're producing batch to batch. So your impurity profile is similar, everything is as similar as possible. But think about those details, but having that representative control to show you run to run is going to be extremely important. So you can trend your assay and kind of get ahead of any problems that you might see if your assay is starting to run into some problems. Definitely open it up to Melissa and to Jennifer or Audrey if you also want to chime in on this. That's a good question.
Nope. It's well done. It's something that actually I think we as an industry and the regulators, the complexity of these new modalities, and so it's probably again, maybe sharing best practices and also sharing the science that goes across that's going to move everything forward. So I think we're in a unique situation because we do get to see a variety of different methods and different assays, so we can share some of those best practices. I know I'm the moderator, but I have to talk, so it's okay. All right, back to the question. I like this one. Let's give this one to Jennifer. How does the regulatory affairs team support a successful AAV gene therapy transfer? But I'm going to broaden that up. Regulatory affairs. With these new modalities, it seems like we're getting a lot of good guidances from that are being issued by the regulatory agency, but then being able to interpret them and execute, implement them into a process. So I'll turn that to you, Jennifer.
Yeah, thank you. So I think from our experience, right, helping different clients to filing their INDs, and also like seeing a lot of information requests coming from the FDA. So definitely the any assay that is used to determine potentially the potency, right, of the AAV, right, as a surrogate in phase 1, right, would be very important, such as like infectious titer. And agency is really focusing on how accurate, right, how can you control the assay, right, so that your assay acceptance criteria or variability, right, is as tight as possible, because that could be used to determine the dosing, right, and the dosing will then end up impacting your dose escalation, as well as the measurements of the success, right, of the therapy. So where we see that is very important is for those assays, right, that really have to work on eliminating any variability, right? So again, what I explained earlier, that you're really limited to factors that are really like either representing the process variability and or the method variability. But method variability we try to minimize, right? And then of course at WuXi we also develop different panel testing, right, for different modalities. So we have known and seen like what are the different required testing, like to indicate like identity, potency, purity, safety, etc. Right. So we have testing recommendation for each category, and we put a modality panel together as a starting point that we help to discuss with clients. So I think those are the really the something that we have seen and work on with experience, that we really think that will be helpful for any clients approaching us, right, to be able to utilize what we have learned, right, with the accumulated experience that we have. So I also want to turn over to Heather to see if she has anything to add.
I think you covered it very nicely. Thank you, though.
Okay, so we won't be able to get through all the questions, but we'll definitely we will respond as we're coming up to time. And before I turn it over back to Abby, I just wanted to share sort of my parting thoughts. I mean, you can see that it takes a village, right? And it takes many different disciplines. And really the key is being able to have all these disciplines work together to have a successful method transfer. So that would be my final point that I think that we wanted to share with this group. So Abby, I'll turn it back to you.
Thank you so much, Audrey, and thank you to all of our panelists today for a very informative and thought-provoking discussion. And thank you very much to Dr. Audrey Trang for doing a fantastic job moderating today's session. That's unfortunately all we have time for, but as Audrey said, any questions that we didn't manage to get to today, we will reply via email. And today's webinar will be available on demand tomorrow, so do look out for an email from us with the link. All that's left is to thank our panelists once more for a great discussion today, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you.